restoration, and environmental pollution management, DNA quantification, molecular identification, enzyme kinetics, cell and tissue culturing, environmental monitoring, and air sampling with environmental and metals analysis. So we would like to welcome as a session chair Dr. Prabhat K. Chauhan, sir. Thank you very much, sir, for joining.
Thank you very much, sir.
Okay. Now we have our invited lecture from Dr. Santosh Kumar, sir, in this international conference. Dr. Santosh is actively involved in teaching and academic activities in the areas of plant biotechnology, microbiology, and agricultural sciences. His broader research interests include plant microbe interactions, biocontrol strategy, and the development of sustainable alternatives to chemical pesticides. He is a researcher in the field of plant pathology and agricultural microbiology with a focus on sustainable crop protection strategies. His research primarily explores the antifungal and antibacterial potential of plant-derived compounds against important phytopathogens. He has conducted extensive work on evaluating the botanical extracts for their bioactive properties and their potential application in eco-friendly disaster management. Through his research and academic contributions, he aims to promote environmentally safe agricultural practices and support innovations that contribute to sustainable crop production. So we would like to welcome Dr. Santosh, sir, with his presentation in this international conference in the track 4. All the best, sir. Over to you, sir. Santosh, sir. Santosh, sir, kindly make your slide in the presentation mode. Sir, your mic is mute, I guess. संतोष सर
माइक आवाज नहीं आ रही इसकी।
Hello, Arpit sir. Arpit sir,
Yes,
can
ma'am.
we start with the candidate till the sir will join? Then
No.
afterwards we will continue the invited talk.
Yes, ma'am. He's not able to join with his PC. He is joining with his mobile right now.
Okay. Ask sir, or please start with the student presentation.
Hello, sir. I am ready. May I start?
Hello, Santosh sir, are you there?
Yes, ma'am. He's just sending me his PPT. I will share his PPT and he will talk like that.
Okay.
I will share it. So he's just sending me his PPT.
Okay.
So
Yes.
I'm just waiting because being an invited talk, we would like to start it from there only.
Yeah. Yes, sir. Yes, sir. Please.
Okay.
Am I audible now?
Yes, sir.
Hello?
Audible?
Okay.
Yes,
You are
sir.
audible.
Is my slide visible as well?
Yes, sir.
Yes.
It's
Okay, thank you so much. Thank you so much for the confirmation, and I sincerely apologize for the delay.
No
Good
issues.
afternoon, everyone. I'm Santosh Kumar, Assistant Professor in Department of Agriculture Science at AKS University. Today, I'm taking pleasure to present a work on botanicals in crop protection, focusing on the potential as sustainable alternative for managing the important plant pathogens. This is actually the demand of the time, and this work actually explores the antifungal activity of plant extracts and highlights their relevance in the sustainable agriculture. See, what the problem as of now is the crop protection is the critical component of agriculture productivity, and we all know that almost 20 to 40% of global crop loss is due to this damaging of the crop by the pathogens or by the rodents and all other things. And the fungal pathogens in particular are the major contributors to these losses. So the need is the developing of effective and sustainable strategies for disease management is now become essential for, we can say, food security as well as stable agriculture production. But what exactly is the requirement? So when we talk about the present scenario, we are gradually using our the chemical pesticides. But these chemical pesticides, although we have attained the food security, we are able to feed people, we have surplus productions, but it has come with some collateral damage, like this thing can lead to environmental contamination. It can lead to loss of beneficial organism. Like we are in our university itself. Whenever we spray some insecticide or fungicide in our agriculture field, we incur the loss of honeybee production in apiary. So this all comes with some kind of collateral damage that we need to focus upon. The excessive reliance on these chemical pesticides has really created many problems. So the major, if you can count, that could be environmental pollution, the pesticidal residue in food, development of resistant pathogen strain, which could be a great threat in coming future, that we may not have any effective pesticides acting against them. And obviously, as I said, that beneficial organisms are also getting impacted on this. Because of these challenges, there is a need, or I would say that it has become the necessity to find some eco-friendly alternatives for plant disease controls. And I present botanicals in crop protection. This thing is not new. It has been a traditional time from prime immortal that we are using these botanical alternatives in our crop control. But if we go in the scientific way and assess the impact and its position, that how it can become beneficial and actually could it be sustainable, then we get the answer yes. Why? Because these are biodegradable, there won't be any residue left, it would be environmental safe, and obviously, like we are talking about the residual effect of pesticide in our food and in our eatables, and that would be generally less toxic to humans as well as animals. And as I said, this is not a new concept. Since time immortal, the farmers, the old people are using this thing like garlic, the neem, the tobacco leaves in controlling their crop and using it in different varieties of way, I would say. Like they prepare dust, they prepare some decoction sort of thing, or they just simply means like soak and spray the water on their crop. So this is not an entirely new concept what I am talking about, but yes, a scientific validation will give it a new path. So, if we try to understand that how these botanicals can be useful to us, so the most common type that is being used includes plant extracts. We all know the plant possesses essential oils; those essential oils can be used. The plant powders can be used. The commercial botanical formulations, that what we are talking about, that what we may have work to move on. So these are the things that we can use in our botanicals. For example, azadirachtin from neem. We are just soaking the neem or boiling the neem in water and just spraying on the crop. Sometimes it works, sometimes it doesn't. But if we isolate the azadirachtin and study the effect of those bioactive compounds, then, and if we are able to formulate the things, then probably things might change. So again, the azadirachtin from neem, pyrethrin from chrysanthemum. Chrysanthemum is emerging as the most guard crop for many of the crops. We are planting chrysanthemum as the guard crop. And various essential oils and aromatic oils that we all know that citronella is world famous for the insect repellent, and the aphid and the white fly, we can get rid of it. So these things are available. We need to understand their mechanism and how to formulate that thing for our scientific purpose and scientific work. So if I may take some time to present that what could be the mechanism of their action. They may show antibacterial, antifungal property. They may be repellent or even feeding deterrent. I'm not just talking about the pathological portion wherein we have pathogens associated. I'm also talking about the carriers. I'm also talking about the rodents that is eventually leading to this crop loss. So they can act as repellent, they can act as the feeding deterrents, and again the resultant will be the pest or insect will be attacking our crop. The chances will be minimum. Okay, so These botanicals often contain multiple bioactive compounds. They can act through several mechanisms. They can have a synergistic effect. One compound may interact with the other one, and cumulatively, they can lead to the control of that particular disease. If we are talking about the fungus or bacteria, or it can act as a repellent. So if I talk about some classic example of the botanicals that have been used is the neem extract. This is the target against aphid, whiteflies, caterpillars, generally used in vegetables. Garlic extract, very much effective against fungal pathogen. Pongamia oil, storage pests like in grains. Some of means after harvesting, we use pongamia oil for mixing with the grains and during the storage of paddy or some oil seeds. Lantana extract. Lantana, I must say, is although we see this as a weed and invasive plant, but it holds enormous bioactive component that is still need to be harnessed for the beneficial use. But yes, it has got excellent repellent property against the field crops. I myself conducted some work on in this field. I tested the Cassia fistula plant against 4 pathogens: the Alternaria, Cercospora, Colitotrichum, and Fusarium. These are some findings, and I must say that the methanolic extract, the way the concentration was used up to 1 milliliter, the methanolic extract proved to be the most effective inhibitor across all phytopathogens. So this work I started as a foundation work wherein I can use for further research in this field for formulating something else. These are the results of Cercospora. And this one is at 3 DAI and 5 DAI. The finding again, we reached up to 75%. In Alternaria, the strongest inhibition in the methanolic content, we reached up to 93.75% of inhibition. In Cercospora, we reached up to 69% of inhibition. In Colitotrichum, this one is slide for Colitotrichum. In Colitotrichum, we reached up to 75% of inhibition, again in methanolic extract. But I must say that the methanolic extract gave a quick result that means very soon it controlled it provided the inhibition. But in long run, acetone extract was much more sustainable in comparison to methanolic extract. Again, the next is of Fusarium. In Fusarium, we got up to 72% of inhibition, and it sustained for another 2 days. After in 5 days also, we got somewhere around 70% of inhibition. So if I try to summarize the experimental finding, if I try to summarize it, I would say that it is again concentration dependent. Like the higher concentration we provide, the more inhibition we got. Now here as through this talk, I just want to invite people who are interested to come forward and do more work in this field to determine the exact concentration. See, I took 3 concentration and I found that 1 ml is very much effective. So I urge people to come forward and take more work in this field, testing different extracts against phytopathogens, so that a complete formulation can be made and that can be used commercially further. Now, if we talk about the outcome, what we can achieve. So we are talking about the sustainability this theme is based upon women empowerment. We all are working, and we all want a minimum damage, high profitability, maximum engagement for the people. So if we have a clear cut formula, a clear cut procedure of preparing these botanicals, then we can take it as a tool for women empowerment. We can set up various help groups so that they can do the production work, and rest can be attained in the field, kind of thing. So, as I said, that again, it comes with some challenges, like we have Ayurvedic system in our clinical, animal clinical science, humans per se. Same way, and there also we find that sometimes it becomes effective, sometimes it may go other way. That challenge is still there. So that's the thing, that to the first thing we need to work here is the standardization and do the quality control. The second thing, we need to work upon the dosage compensation, the understanding of the mechanism of action, and again some toxicity and other residual work need to be done. But if we talk about it's like opportunities, yes, opportunity lies. If these things work, we can set it up as a startup for the new botanical verticals. We can have an expansion and wide area of research. So this holds a promising discussion. So I would like to conclude this talk with the findings that what I have got with my work. I would say that botanicals really hold some good promising future. And again, the future perspective is very much available. More and more research can be done in this field. And again, we can come out with certain good product that can be used in controlling our crop damage. So with that, I conclude my talk, and I thank the authorities for giving me opportunity for presenting my talk. And I am open to take any questions if you have.
Thank you very much, Dr. Santosh sir, for a nice presentation on botanical in crop protection as alternatives of pesticides.
Thank you so much, sir. Thank you.
Thank you, sir. Sir, you can disconnect the sharing option.
Sure, sir.
Okay. So now we are moving towards the technical presentation sessions. And for that, I would like to welcome Mr. Pankaj Kumar Gautam with his title, An Efficient Sentiment Analysis Framework Using TF, IDF, and GRAM Features.
Thank you, sir. Am I audible?
Yes, sir, you are
Yes,
audible. Just present your slide.
Okay, thank you, sir. Thank you.
Prabhat sir, please, if you
Yes,
have any
okay.
question after the presentation, you can ask. Thank you.
Okay. Kindly start your presentation, sir.
I am visible.
Yes, sir, your slide is visible. Go ahead. Start from first slide.
Good afternoon, everyone. I am Dr. Pankaj Kumar Gautam, Assistant Professor in Department of Computer Science at Sharda Mahavidyalaya, Sallanagar. I am going to present my research topic on an efficient sentiment analysis framework using TF, IDF, and GRAM feature, hybrid feature selection, and Actuna hyperparameter optimization. These are the outline of my work. Introduction. Sentiment analysis is basically the branch of natural language processing where we have to extract the opinion or emotions of user given reviews from various social media platforms. Since these reviews are very huge in amount, so manual analysis of these reviews are not possible. So there have a need of an automated process which classifies these reviews in appropriate class. The motivation behind my research is the rapid growth of online reviews and social media tests. Since day by day our social media are increasing, so there have a huge amount of user opinion about any service or product. Organizations and various service provider need to analyze these reviews to know the customer opinion so that they can improve their product quality or service quality. So there have a need of an accurate automated sentiment classification process to represent these textual data in a suitable format so that machine can understand and optimization technique classify into appropriate classes. Since our data is in a textual format, so there have a high dimensional feature space. And since high dimensional feature space are present, then there may be a possibility of some redundant and irrelevant feature. So these irrelevant and redundant feature are need to be removed. So many we have a need some technique. Manual hyperparameter tuning, which is already used in the previous work, is inefficient because this is time consuming and complex. So there have a automatic framework for better classification performance. These are the objectives of my research to develop an efficient sentiment analysis framework, apply TF-IDF with n-gram feature representation, use hybrid feature selection using one-way ANOVA and recursive feature elimination wrapper method, apply Optuna hyperparameter optimization technique, which is an automatic hyperparameter selection technique, which reduces the complexity of traditional hyperparameter optimization technique. Various classifiers are used to evaluate the performance of the model. These are the methodology: data collection, text preprocessing, feature extraction using TF-IDF, n-gram representation, hybrid feature selection using one-way ANOVA and recursive feature elimination method. Model are evaluated using various classifiers like LR, KNN, SVC, DT, GNV, and RFC with stratified 10-fold cross-validation and hyperparameter tuning using Optuna. Our data set is IMDb movie review data set, which is obtained from the Kaggle platform. And this data set is openly for any research work, which contains 46,173 user rating, in which rating column is mainly assigned to score. Our data set contains 7,740 positive reviews, 1,344 negative reviews, and 916 neutral reviews. Then text preprocessing is performed on the textual data to remove punctuation mark, special character, stop word, and then tokenization is performed on the textual data to convert into word format so that it can be used by the feature selection process. TF-IDF is used to extract a feature and converts these textual data into vector formats so that machine can understand these data. Here, n-gram representation is used to capture the contextual relationship between the word, where we have used unigram and bigram in our experiment, where unigram represents single word and bigram represents the pair of consecutive words that make a phrase which can be informative for our research. Like very good, not bad is a bigram example. Hybrid feature selection method reduce the dimensionality of the textual feature. Then ANOVA basically is a statistical filter method that select a feature from the previous stage on the basis of F value. Then recursive feature elimination method remove less important feature, and this hybrid approach select the most relevant feature which are essential for our experiment. Hyperparameter optimization. Basically, previous study, manual hyperparameter optimization is used where it takes time and too much complex process. So here we have used Optuna based hyperparameter optimization technique, which is automatic hyperparameter selection technique that select the most appropriate hyperparameter for our classifier. Logistic regression, KNN, SVC, decision tree, Gaussian naive Bayes, and random forest are the classifier used in our experiment to assess the classification performance. Here, tenfold cross-validation technique is used with stratified technique to maintain the same class distribution as available in the user given data, so that the biasness can be removed and the our result will be reliable. These are the accuracy, precision, recall, and F1 score performance matrices used to evaluate sentiment classification model. Our experiment, we have used many feature selection method like one-way ANOVA plus recursive feature elimination, mutual information with 10% feature, 20% feature, 30% feature, and 50% feature, genetic algorithm, principal component analysis, and in another scenario, we have perform experiment without feature selection. In both cases, with feature selection method, one-way ANOVA and recursive feature elimination, KNN classifier got the highest accuracy of 95.10%. With the same combination, F1 score for the one-way ANOVA plus recursive feature elimination method is highest in KNN classifier, which is 92.80.
Pankaj sir, kindly conclude your talk. Your time limit has been extended.
Okay, sir. With conclusion, I can say that proposed framework integrate TF, IDF, n-gram hybrid feature selection method and Optuna optimization give us a complete framework which is able to reduce the dimensionality of the feature data set, improve classification performance, and can be applied to other sentiment classification domain. Further, it can be used with deep learning method and more large data sets, more advanced embedding method like word2vec and BERT can be used on these framework for further work. These are the bibliography of our work. Thank you.
Thank you very much, Dr. Pankaj Kumar. Can you explain about the errors of your analysis?
Basically, sir, I have analysis on the basis of differences given by the various classifier in performance matrices like accuracy, F1, precision score, and recall.
Okay. And what is the accuracy of this sentiment analysis?
95.10%, sir.
Is that accurate, sir?
Yeah, it can be enhanced with more technique like word to vec method and a deep learning method, but which can be used in our future works.
Okay. Thank you, Dr. Pankaj Kumar, sir.
Thank you, sir. Thank you.
And next we have Nilesh Srivastava, sir. Please join with your
Yes, sir.
presentation.
Yes, sir. Yes. Is the screen is visible, sir? Hello.
Sir, your slides are not visible.
Slide is not shown, sir. And my voice is audible, sir? Yes,
Your voice
yes.
is audible. Your voice is audible.
Just. Is it visible, sir?
Yes,
Hello.
it is visible, but we are not able to see your slides.
Hello, sir. Can you see the
Just
slides?
click on sharing option.
I clicked on the sharing option only.
Bring the call back here. Bring the call back here. Click on that. It is written there. Bring the call back here.
Yes, sir. Bring the call back here. Yes, sir.
Just click on that. Now go to your PPT. First of all, go to your PPT. Where it is on your. Just minimize the Google. Go to your PPT.
Is it visible, sir?
No, it is not. Click on stop presenting. First click on stop presenting.
Yes, sir. And, install.
Just first open your slide. Sir, पहले अपनी slide open करके रख लीजिए.
So slide is open at the ground.
हाँ, अब आप शेयरिंग ऑप्शन पे क्लिक करिए. शेयर ऑप्शन पे जाके शेयर इंटायर स्क्रीन पर क्लिक करिएगा.
Yes, sir. Again, I have shared. Is it visible?
No, not yet. Okay, now click on the PPT. Now click on the PPT which is given below.
Hello, sir.
Sir, still the slides are not present. Take your time. Just leave the Google Meet and join it again. Sir, पहले इस बार leave करके फिर से join करिए. Okay, now it is good. It is visible, sir.
It is visible.
Yes, it is visible. Just go ahead.
Uh, Kindly start from first slide, sir. Yes.
Okay, yes.
Yes, sir.
You are good to go.
Good, sir. Yeah, good afternoon, everyone. I am Nilesh Srivastava, and the topic to present is the auto-scaling framework for workload-aware resource allocation in cloud computing. And points which I am going to cover during my presentation are the introduction, need and significance of the research, hypothesis, review of literature, proposed methodology, expected outcome, and references. Cloud computing I have chosen because cloud computing is a distributed computer process that provides basically on-demand access to share computing resources. Means to say that when randomly any process comes and if it requires any kind of I can say resource allocation, then cloud computing helps in doing the process efficiently. In this framework, actually, I have used novel assembled based workload aware auto scaling process, which I will describe later. So in cloud computing, actually what happens, server, storage and networks and various other application can be the process of allocation can be performed through the internet. If I go to the introduction part, cloud computing is the backbone of the modern digital service. Nowadays, we can see that everywhere we are using cloud for storage. And because of the scalability, that means to say that to increase the performance accordingly, I am using this cloud computing platform for my auto scaling framework for allocation of the workload. Moving towards the next slide. About the auto scaling framework, it includes rapid growth of cloud-based application with dynamic and variable workloads, inefficiency of static resource allocation. Actually, auto scaling is basically of three types: horizontal scaling, vertical scaling, and hybrid scaling. In horizontal scaling, what happens? We can add or remove virtual machines or various types of containers. Whereas in vertical scaling, we can increase or decrease the resources like which are existing in the machines. Now coming to the hybrid one, it is the combination of both horizontal and vertical scaling. Limitation of where we are using CPU, various memories for storage and other networks. My objective is to maintain the quality of service and service level agreement, SLA, which reduces the resources over provisioning and under provisioning. Also helps in optimizing the cost and energy consumption. Why there was the need of such kind of framework, means which I'm going to explain the auto scaling framework. The need became during the research, it came into existence that sometimes what happens, exact and proper workload resource is not allocated. Means as we know that there dynamically changes to send using the data. And for that purpose, resource allocation is required. Here you can see that in the need I have written, existing auto scaling techniques are mostly static or threshold based, which are inefficient in handling highly dynamic and unpredictable cloud workloads. There are also critical need for workload aware in and intelligence resource allocation mechanism to reduce resources wastage while ensuring the quality of service and service level agreement compliance. I'm audible, sir. Hello. Hello.
Yes, sir. Yes,
Yes,
sir. Proceed, proceed, sir. Proceed, sir.
Okay. Let me proceed it. Thank you. Okay. Thank you, sir. In the hypothesis part, when the proposed in novel ensemble significant improvement. So for that, I have used novel ensemble computing enhanced resource management by combining multiple learning models to improve the accuracy and reduce the service level agreement, which was not in the primary hypothesis. And that creates a major and important role in the auto scaling framework for workload aware resource allocation in cloud computing. Various review of literatures, various literature which I have used during this research are mentioned in the screen. These are the latest literature of the year 2024, 23, and 25. I have taken into consideration, and in the proposed methodology, I actually a Jupyter notebook have been used, from where it is the open source browser from where interactive environment are used to create documents. And here I machine library, which helps in providing the data collection and pre-processing, algorithm implementation, predictive ensemble model development, workload aware resource allocation, evaluation of ensemble model, and validating and testing. The expected outcomes you can see here are the predictive workload forecasting, proactive resource allocation, improved utilization, and the basic thing, which is the quality of service, has been enhanced along with the reliability, and the major factor, which is the cost efficiency, is also being cost is also reduced. Various references are here. Thank you, sir.
Thank you so much, sir.
Thank you very much, Nilesh sir, for your nice presentation on cloud scaling. Can you explain about the role of cloud computing in scientific research?
Your voice is not clear. Pardon, please pardon the question.
What is the role of cloud computing in scientific research?
Actually, it allows user to scale the resource in dynamically as the need. Yes.
Yes. And another is, you have mentioned here, is the two types of hypothesis, that is a primary and alternate hypothesis. Are you here know about the null hypothesis and alternate hypothesis?
No, sir.
Or you understand about both null and primary hypothesis and alternate hypothesis? What is the difference between null hypothesis and alternate hypothesis?
So
During a research
yes, sir. The alternate.
What is the difference between null hypothesis and alternate hypothesis? H0, H1 in the research.
Yes, sir. During the null hypothesis, the question which came for the enhanced version or the proposed methodology, that
नहीं मेरा मतलब ये
find
है
out, and that for that we have to make some significant improvement. Yes, sir.
नहीं मेरा ये कहने का मतलब है कि जो नल हाइपोथेसिस होता है, किसी रिसर्च में नल हाइपोथेसिस जब रिजेक्ट होगा तभी तो अल्टरनेट हाइपोथेसिस एक्सेप्ट होता है. इसलिए तो सिग्निफिकेंट आपने कहा, सिग्निफिकेंट इंप्रूवमेंट यहाँ लिखा भी है आपने.
यस सर, यस सर.
हाँ ठीक है, ओके, थैंक यू वेरी मच निलेश सर,
यस,
फॉर योर नाइस प्रेजेंटेशन.
So for our next presentation, we have Diksha Chaudhary. Diksha Chaudhary,
Yes, sir.
kindly share your screen with presentation.
Okay, sir.
Nilesh sir, kindly log out from the sharing option. Thank you.
Yes. Yes. Yes.
Yeah.
Sir, hello.
Hello.
It is audible,
I'm
madam.
audible.
Your
Yes,
screen
it is audible. The screen is not perfectly.
your half screen is only visible. Just don't just move your mobile in the vertical direction again.
Okay, sir. I try.
Okay, Diksha, do one thing. Hello.
Yes, sir.
Just send me your slide. I will present from here and you will just dictate it. I will present it because on mobile it is not visible at all.
Okay, sir.
Just send me your PPT. Do it first. Just log out from this screen sharing option.
Sir, I send it.
Prabhat sir, is it visible?
Yes, sir. It is perfectly visible.
Okay. So
Sir, can I?
Diksha, start talking. Start presenting your work.
Good afternoon, everyone. My name is Diksha Chaudhary, and I am currently pursuing my post-graduation in MSc Microbiology from Department of Biotechnology, AKS University, SATNA, MP. Before we start, first of all, I would like to sincerely thank all respected organizers, respected experts, and also my guide, Mr. Paras Kose sir, for guiding me on my research and give me this opportunity to present my work in front of you all. Today I am going to present my research on biodegradation of plastic by microorganisms which isolated from open dumping site. This study focuses on identify plastic degrading microorganisms and exploring eco-friendly solution for plastic waste management. The abstract is plastic pollution is a major global environment problem due to its persistence of synthetic polymer in ecosystem. Conventional disposal methods such as incineration, landfilling cause environmental hazards. Microbial biodegradation offers an eco-friendly approach to degrade plastic waste. This study focuses on isolating microorganisms from open dumping sites and evaluating their potential for plastic degradation. The research aims to identify efficient microbes capable for breaking down plastic polymers and contribute to sustainable waste management strategy. The introduction is, sir, second slide.
Diksha, just tell what you have done in your research, okay?
Okay, sir.
You have less time and make it fast.
Okay, sir, I try. My objective of study to collect soil sample from open dumping site to isolate uh, microorganisms which is capable for degrading plastic, to screen and identify efficient plastic degrading microbes. Fourth is to evaluate the biodegradation efficiency of the isolated microorganism. And next is methodology is first the sample collection and analysis. Soil sample will be collected from selected open dumping sites and analyze their physicochemical properties like pH and moisture level. Second is isolation of microorganisms. Microorganisms will be isolated using a serial dilution and culturing techniques on appropriate media. And these are the pictures during my research. And this is a pore culture pouring, and these are streaking. And next is screening of plastic degradation. Isolation will be screened using a minimal salt media containing plastic as the sole carbon source. And fourth is biodegradation study. Plastic degradation will be assessed by the weight loss measurement, surface changes, and microbial growth analysis. Fifth is characterization of efficient isolates. Morphological, biochemical, and basic molecular characterization will be performed. Expected results of this research will be isolation of efficient plastic degrading microorganisms. Second, reduction in plastic weight after a microbial treatment. Third, surface degradation observed under scanning electron microscope. Fourth is chemical change detected. Their finding will be confirmed microbial plastic degradations. And next is conclusion. The present study highlight that potential of microorganisms isolated from open dumping sites in the biodegradation of plastic materials. The microorganisms produce enzyme like oxidase, peroxidase, which is capable for breaking down complex plastic polymer into a simple compounds. Therefore, microbial biodegradation represents an environmentally friendly and sustainable approach for reducing plastic pollution and improving waste management strategies. Thank you for all your kind attention. I will be happy to answer any question. Thank you.
Thank you very much, Diksha Chaudhary ji,
Yes,
for
sir.
nice representation on biodegradation of microplastic.
Yes, sir.
During the plastic isolation of microbes, you have used the serial dilution methods. What is the dilution factors?
Sir, dilution factor from 10-1 to 10-9.
10-1. 10-1 is the very high concentrated microbial. How do
Sir, I prepare between 10-1 to 10-9 dilution and use 10-4, 10-6 and 10-7 dilution for my serial dilution.
Okay, thank
Yes,
you. Which methods are used for the identification of molecular analysis? किया आपने उसमें?
No, sir, not now.
तो फिर कैसे आपको ये आइडेंटिफाई हो गया कि ये हमारा बैक्टीरिया जो है वो या माइक्रोब्स जो है हमारा बायोडिग्रेडेशन में हेल्प कर रहा है?
Sir, I'm continue this research under my guide, Mr. Paras Kose, sir. I am working on this, sir. This is not an expected result. We are hope to. Sir, I'm
आपने जो
currently
you have identified a specific type of microbes which have a special characteristic for the degradation of microbes, which only explain about the special characters of your microbes.
Sir, Not, I am not, sir,
It is a बोलिए.
sir, actually, I am not working on the identify microbes. I am working in the soil sample, and which I don't know which microbes is present on the soil sample. I am working. I'm just thinking there are something microbes will be identified after during my research work, sir. Sir, I am continuous. Sir, currently I am working.
Okay.
Okay, sir.
ठीक है. Thank you very much.
Thank you, sir.
Thank you, Diksha. Now we are moving towards our next candidate, Mr. Himanshu Pandey. Please be ready with your presentation. Share
Okay,
your
sir, I
screen.
am audible.
You are audible. Just share the screen of your presentation. And just stick to the main research part, whatever you have done, right?
Sir, screen is full.
Yes, it is visible. Just stick to the whatever you have done. Okay.
Okay, sir. Good morning, respected chairperson and honorary judges and the participant present here. First of all, I will be good. Thank you, organizers for first international conference with research innovation with women employment with sustainable agriculture development is given opportunities with present research work. My name is Himanshu Pandey and today we'll be presenting you research title Production Biodiesel using single cell oil SCO technology. The project work will be guidance will be Dr. Ashwini Eva and my co-author with Sahida Wadwani. Problem will be no is the increasing population be continuously increasing the dependence for the fuels with creating the CSV environmental problem such as global warming, air pollution, and health issues with including respiratory diseases. There is very important with sustainable eco-friendly
Himanshu,
fuels.
your slides are not moving. Your slides are not moving. You are still on the first title slide. Just move it ahead. Your slides are not moving. Just reverse it from presentation mode and click on the second slide. Don't make it on the presentation mode. Just share it again and click on entire screen. Still, it is not visible. Oops. Stop sharing. First, stop sharing and reshare it again. And this time, click on entire screen and then go to your PPT just.
Okay, sir. So slide is visible.
Yes, it is visible now.
Okay.
Start from objectives.
Implementation of the current biodiesel. Biodiesel is a constantly on the promoting as an alternative because it is a renewable and environmentally friendly. Studies suggest with the biodiesel can reduce particular matter emission will be about 47% and reduce hydrocarbon emission will be nearly 62%. produce compulsory less smoke and during combustion. Biodiesel is an alternative and conventional fossil fuel. Limitation for the current biodiesel. Current biodiesel will be presenting the mainly dependent all the patient will based EPS oils and created between food and fuel resources. And additional high protection and cost limitation, scalability, and also challenges. There will be necessary for the explore and the sustainable sources of protection. Problem solution. Oil is used a potential alternative. Single cell oil is microbes, lipid production by microorganisms, by bacteria, yeast, and fungi. This is the microorganism oleaginous properties, meaning they are the accumulate high amount of the lipid. Inside there are the cells which can later and can convert into the biodiesel. Role of the single cell oils. Single cell oils is the provide several advantage microorganisms to accumulate the high intercellular lipid content. And will be independent with climate seasonal changes and agriculture waste can be used for the substrates support circular bioeconomic waste and utilization. In methodology, microorganisms are cultivated under level conditions. They carbon substrates with promote the lipid accumulation. They are lipid converted to the biodiesel through transpiration and fatty acid, ethyl ester. With key and organisms, your study will be how Bacillus subtilis microorganism. Their bacteria is a safe, well-be studies with easy cultivation and capability with the using agriculture and oil wastes. There will be a providing a promotion across the stable biodiesel production. Conclusion: single cell oil is a technology potential contributes significant renewable energy production. It is the help of the biodiesel production, but also support waste utilization and environmental stability. Single cell oil technology has the potential and contributes significant renewable energy production with agriculture based management and reduce environment pollution. We are not biodiesel developing sustainable energy. Solution that we integrated waste management, environmental protection, and renewable fuel protection. Thank you. And attention, be happy and answering these questions.
Thank you very much, Himanshu ji, for your nice presentation on biodiesel. Can you explain about the calorific value of biodiesel and fossil diesel? What is the difference?
So this is high saturated lipids to biodiesel products, quality lipids. Like palminic acid.
I am asking to you,
Yes.
what is the calorific values of biodiesel as you presented here and normal fossil fuel, that is a petroleum? What is the differences? How many? Whose calorific value is more higher? Are you understanding what I'm saying? Okay. Okay, Himanshu.
Yes. So I don't understand it.
ओके मतलब मेरे कहने का मतलब ये है कि जो बायोडीजल आप बना रहे हैं, ठीक है, अच्छी बात है ये।
Yes, sir.
क्या उसकी कैलोरीफिक वैल्यू जो है, जो हम फॉसिल फ्यूल यूज करते हैं, उसी का अल्टरनेट आप बना रहे हैं ना?
Yes, sir. It is a similar.
ऑलमोस्ट सिमिलर
Yes, sir. It's
or 100% similar?
actually, sir, this is research is already is done, but it's not a pilot scale working.
ओके वो सही है, वो सब सही है कि पहले रिसर्च हो चुका है, आप उस पर काम करेंगे, वो ठीक है। मैं कॉमन क्वेश्चन पूछ रहा हूँ कि क्या कैलोरीफिक वैल्यू दोनों की सेम होती है या डिफरेंस होता है?
sir, I told you.
ओके कोई बात नहीं. ओके थैंक यू सर.
Thank you, Himanshu. Just stop your screen sharing option.
Okay, sir.
And next we have Shreya Vishwakarma. Shreya Vishwakarma, start sharing your slides.
Yes, sir. That's good. Sir, my screen is visible.
Your screen
Yes, it is perfectly
it is visible. Just go ahead and stick to the whatever you have done. Okay.
Yes, sir. Good afternoon, everyone. My name is Shreya Vishwakarma, and I'm currently doing work on plastic production with microalgae. Plastic productions are now majorly environmental causing problems that contains severe diseases, and they doesn't dissolve in thousands of years. That's why we produce bioplastics, which is made up of microalgae. Microalgae, these are lab techniques which where we separates different types of algae. We found 5 types of algae, that is cholera vulgaris, that is algae where I am currently doing my project. And this is lab techniques where I am studying different types of algae to found different types of lipid content, carbohydrate content, and protein contents which is found in algae. They are lab techniques where I am growing large scale in labs, and these are methods and material. In algae production, we use BG11 media for the nutrient growth of microalgae. In there, we contain nitrogen, minerals, and trace elements where the microalgae growth properly. In this, we found a biological analysis on spectrophotometer where protein content in 540 nanometers and carbohydrate analysis from 490. Here I found lipid extraction from orange peel biomass, where I use after this algae biomass lipid content. These are the difference of normal plastic and bioplastic, which I have working currently now. And normal plastic have 400 to 500 years to degrade, where a comparison that bioplastics are degrade in monthly or yearly. Microalgal biomolecules use plastic productions stress. They are conditions for increasing of PHB accumulation, biomass content protein or lipid, where precursors of bioplastics and components are found in microalgae. These are some reference where I obtained microalgae production, microalgae with bioplastic. Thank you, sir.
Thank you very much for your presentation, Shreya Vishwakarma, on plastic production by microalgae.
Microalgae, yes, sir.
Can you explain about the microalgae production media? Which type of media you are used for the growing of the microalgae?
Sir, I use BG11 media for growth of microalgae.
क्या कंपोजिशन बता सकते हैं उसका?
Yes, sir.
Please.
Sir, sodium nitrate, dipotassium, magnesium sulfate, calcium chloride, citric acid, ferric ammonia citrate, EDTA, sodium carbonate, trace element और distilled water.
मेथोडोलॉजी किसकी मेथड्स यूज की थी आपने?
सर
मेथड्स किस मेथड से आपने किया था? किसी जैसे मान लीजिए यादव एट al 2025 या इस तरह के कौन से कॉमन मेथड्स हैं विच आर यूज इन फॉर ग्रोइंग ऑफ द माइक्रो एल्गा?
सर कॉमन मेथड्स
स्पेसिफिक बताइएगा क्योंकि मेरा भी काम इसी पे है।
सर स्पेसिफिक
जो कॉमन यूज होता है आपके यहाँ, जैसे कोई भी हो गया, ठीक है कोई बात नहीं, ठीक है कोई दिक्कत नहीं है। क्या ये एक माइक्रो एल्गा जो प्लास्टिक प्रोड्यूस करता है ये प्रॉब्लम नहीं होगी बाद में? पहले प्लास्टिक कहा गया कि प्रॉब्लम नहीं है, प्रॉब्लम हो गई, अब इसको हटाने की बात हो रही है, बाद में इसको भी हटाना पड़े तो फिर क्या कोई प्रॉब्लम ये हो
सर
सकता है
ये
इसमें?
तो सर इट कैन डिग्रेडेड इजिली, तो इससे तो कोई प्रॉब्लम नहीं होगी ना, क्योंकि सर ये नेचुरल से इफेक्ट नहीं करेगा।
अच्छा, अभी वर्तमान समय में अंडरस्टैंडिंग ना हो इतनी ज्यादा हमें इसके बारे में,
यस सर,
बाद में जो है ये अफेक्ट करने लगे। इको फ्रेंडली है, मैं मानता हूँ इस तरह से, भाई माइक्रो एल्गा से बनाया गया तो इको फ्रेंडली ही होगा, जाहिर सी बात है। लेकिन जितने भी कंपाउंड्स निकाले जा रहे हैं, चाहे सिंथेटिक हो चाहे किसी भी चीज से प्राप्त हुए हों, वो कहीं ना कहीं अफेक्ट करते हैं एनवायरमेंट को। तो हो न हो ऐसा ही हो सकता है कि ये भी प्लास्टिक जो है बाद में डिग्रेडेड मतलब इम्पैक्ट करे एनवायरमेंट को, ऐसा कुछ है जानकारी आपको?
नो सर।
ठीक है, ओके, थैंक यू।
थैंक यू सर।
Shreya, just stop your screen from sharing option. And now we would like to call Mr. Avinder. Mr. Avinder, start presenting your slides.
Yes, sir. Yes, sir, I am visible.
You are visible, just click on your PPT.
Yes.
Okay, okay. Just drag it slightly so that we can see. Yeah, okay, right, good. Okay. So this is your poster, right? Prabhat,
Yes,
sir. Now we have oral presentations are over. We are moving towards the poster presentations, e-posters. Prabhat, sir.
Okay, okay, sir.
Okay, Avinder, you can carry on.
Okay. Good afternoon, everyone. My name is Avinder Singh. I am doing masters from Punjab Agriculture University in Master of Genetics and Plant Breeding. Today, I am excited to share our research on how sugarcane clones cope with the major challenge, waterlogging. First, we move to introduction. As we all know, the catastrophic floods of 2025 in Punjab caused severe and prolonged waterlogging. This is a huge threat to sugarcane, a critical cash crop for the region. When a sugarcane plant is submerged for a long time, its root can't breathe. This disruption in respiration leads to what we call oxidative stress. Think of it as the plant version of inflammation. It also causes the degradation of important pigments like chlorophyll, which the plant needs for photosynthesis. So our central question was, how do we find sugarcane varieties that can survive this? The answer lies in the plant's chemistry by looking at specific biochemical markers like ones listed on my poster. We can actually fingerprint a plant's tolerance strategy. The ultimate goal of this study was to screen 5 elite sugarcane clones and identify the most resilient ones for future climate adaptive breeding programs. Now we move to material and methods. To do this, we set up an experiment at the PAU Regional Research Station in Kapurthala. We took 5 elite sugarcane clones and grew them under 2 conditions. First one is a control group with normal irrigation and a waterlogged group where we submerged the plants for a prolonged period of 75 days. After those 75 days, we collected leaf tissue samples and analyzed them for key biochemical indicators. First one is proline, an amino acid that acts like a stress shield, helping cells retain water. Second one is chlorophyll, the primary pigment for photosynthesis. Its levels tell us how healthy the plant is. Third one is carotenoids, accessory pigments that also act as powerful antioxidants, mopping up harmful molecules. And fourth one is catalase, an enzyme that neutralizes a specific type of cellular toxin. so high activity can indicate high stress. Now we move to results and discussion. Now let's look at what we found. The data revealed a really clear hierarchy of survival. Let's walk through the clones from the most vulnerable to the most tolerant. The most vulnerable clone is COPB 11. First, let's look at COPB 18 to 11. The story here is quite dramatic. In figure 2, you can see its chlorophyll content has plumped compared to the control. It's the lowest of all the clones. The same is true for carotenoids in figure 3. But the most telling sign is figure 4. Look at its catalyst activity. It's through the roof, the highest among all clones. This is a classic distress signal. The plant is under such intense oxidative stress that it is desperately trying to produce this enzyme to combat the damage. This tells us that the COPB 18 to 11 is highly susceptible to waterlogging. And now we move to the moderate performance. COPB 181 and 18214. They show a mixed response. In figure 1 and 2, their pigment and proline levels are somewhere in the middle. They are showing measurable signs of stress, but their physiological systems are still functioning. They have a moderate tolerance. Now the move to the strong performance 23 to 11. This clone showed a really balanced and effective biochemical response. It accumulated a high level of proline, which is great for osmotic balance, and most impressively, it retained the highest carotenoid content of all clones. This is a key finding because it shows very efficient antioxidant defense system at work. Finally, the clone that stood out as the most tolerant was 18 to 13. Under the waterlogged condition, it accumulated the maximum proline content of any clone. At the same time, it maintained the highest levels of chlorophyll. This combination is the hallmark of a robust protective mechanism. It's effectively shielding its cellular structures and photosynthetic machinery from damage. 18 to 13 is the clear superior performer from all these 5. Now we move to conclusion. So to wrap up, after 75 days of waterlogging, we identified a clear gradient of tolerance with superior pigment retention and proline accumulation, and also excellent candidate with a strong balanced biochemical defense, particularly its antioxidant capacity. On the other end, 18 to 11 showed severe vulnerability, serving as a sensitive check. These biochemical fingerprints are more than just data points. They provide us with a scientific roadmap. By identifying these inherent stress-tolerant traits, this research is a vital first step. It gives breeders like us the knowledge to select and breed hardier sugarcane varieties, ensuring our agriculture future can better withstand the climatic challenges that lie ahead. Thank you for your time. I did happy to answer any questions.
Thank you very much, Arvinder Singh sir, for your nice presentation on waterlogging is tolerance and climate change resilience in sugarcane. Can you explain which types of variety are used in your research work? Either normal variety or local variety or hybrid variety?
Sir, they are hybrid varieties. Tested in waterlogged clones which performed better in waterlogged conditions in the area between Beas and Satluj river.
Okay. Are you applied the normal varieties like local varieties? Only for hybrid variety? You have used for only hybrid varieties, yes?
Yes, sir.
Do you understand?
Yes, sir, I am understanding. Like the field is, I have grown 33 varieties actually in the field, but from the 33, after 75 days, these 5 varieties are performing somewhat better. So I selected these 5 varieties to run the biochemical markers on them.
Okay.
Yes.
What is the changes after your stress condition? What is the changes in your varieties, your treatment varieties, as compared to control? This type of changes you have observed.
Sir, observed that the first one is, as they are waterlogged, their roots can't breathe. So that's why their photosynthetic activities also lose, because the leaves get dried up due to waterlogging stress, and their cane length and diameter also reduced, and some varieties lodged off as they topple down over due to tissue that shrink at the bottom of the cane.
Okay, what is the common? You have used your research paper for the as a methodology for the evaluation of chlorophyll, proline, and so on. Can you explain about the? Can you use Ernon methods for the extraction of chlorophyll? Are you
Sir,
clear?
yes, I am understanding your thing, sir. For chlorophyll and carotenoids, it is a same method of for centrifugation. For centrifugation, chlorophyll and carotenoids, we collected the leaf sample and centrifuge for collectively for the chlorophyll and carotenoids.
Your procedure is good, but I want to which type of procedure you follow.
As in what, sir? I am not.
Okay, sir. कोई बात नहीं.
Sir, same as I mentioned in my poster, I given two references. I followed that procedure. Yes, sir.
Okay.
I somewhat SN based procedure.
Arvinder Singh, I have observed that your procedure is only taken from any research paper. That is not actual. Have you seen about the chlorophyll methods are actually used or procedure are used for the Ernon methods. That is a common methods and scientific proved method. It is not a modified method. You have used only. Okay, your procedure is good. I'm not asking about it.
Yes, sir. I have done under biochemical specialist at our university.
Okay.
Yes, under their guidance, I have performed these experiments. Yes.
Okay, good.
Yes.
Arpit sir.
Thank you, sir. And we have one more candidate, Sumer Singh Rajput.
Yes, sir.
Just present your slide.
Okay, sir. Good afternoon to everyone. Sumer Singh Gehlot. Currently, I am pursuing my PhD degree at Department of Agronomy, Anand Agriculture University, Gujarat. Here, my poster presentation title is Enhancing Yield and Nutritional Biofortification of Finger Millet plus Soybean Intercropping through Optimized Bioformulation in Organic Farming. Introduction. Finger millet is third most significant nutritional cereals in India after sorghum and pearl millet, which contain beneficial minerals like calcium, iron, and zinc, and also vitamins. And it accounts for 85% of all minor millet production in the country, and covering around 1.01 million hectares with an average productivity of 1747 kg per hectare. Soybean, also called as wonder crop, which contain protein level around 40 to 43% and oil content 20 to 22%. In India, it occupies an area of 30 million hectares with the production of 12.04 metric ton and productivity of 930 kg per hectare. Finger millet plus soybean based intercropping system is the most sustainable viable natural ecosystem because the initial slow growth of finger millet which facilitates the better establishment of intercrop. And soybean also can increase the soil fertility by fixing 50 kg nitrogen per hectare. So excessive use of chemical fertilizer lead to cause different type of problem in environment and as well as in soil. So we use organic and on farm produce product for a sustainable production. Here is objective for my research is to study the effect of bioformulation on yield and nutritional quality of finger millet plus soybean intercropping under organic farming. During my research, I take my experiment at College Agronomy Farm, Anand Agriculture University, Gujarat during Kharif 2024. I would take on total 10 treatment and 3 application. In treatment 1, taken only control in which contain only recommended dose of fertilizer, and remaining T2 to T10 use the different level of Jivamrit plus Jivamrit. At sowing time, taken Jivamrit level taken 1000 kg per hectare, 1500 kg per hectare, and 2000 kg per hectare, and Jivamrit level taken 500 liter per hectare, 750 liter per hectare, and 1250 liter per hectare. Based on my study, result revealed that application of Jivamrit at the rate 2000 kg per hectare plus Jivamrit 1250 liter per hectare registered significant higher yield in 68.22 quintal per hectare straw yield and 26.43 quintal per hectare yield of soybean, and record 15.91 grain yield in and 25.24 quintal per hectare yield in soybean. And also this treatment Jivamrit at the rate 2000 kg per hectare and Jivamrit at the rate 1250 liter per hectare register higher nutritional content, nutrient content like nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, and as well micronutrient like phosphorus and zinc also register highest in this treatment T10. And this treatment T10 also superior in soybean as well. Nitrogen, phosphorus, and potassium content highest in treatment T10. So conclusion. Application of Jivamrit at 2000 kg per hectare plus Jivamrit at 1250 liter per hectare at sowing, with successive 4 application of Jivamrit at each 20-day interval in finger millet plus soybean intercropping system under organic farming, performed to be the most effective and outperform in achieving grain and straw yield and nutritional quality of finger millet plus soybean intercropping. Thank you, sir.
Thank you very much, Sumar Singh Rajput, sir.
Okay.
Can you explain which type of pot experimental treatment are used in your research?
Sir, yes, sir.
Yeah. Yes, please explain.
Sir, in T1, we just take control. In control, we take only recommended dose of fertilizer. And rest of treatment T2 to T10, we give a seed treatment of Bijamrut, and at time of sowing give a Jivamrit and Jivamrit and different level.
Okay, let's see. It's okay. You have used your CRBD design.
Okay, RCBD, sir.
Complete randomized design.
Yes, randomized complete design.
Design. What is the CRBD? Seizure of CRBD.
What, sir?
How to explain about the CRBD design?
CRBD is mainly used in lab study, where fertility is experimental unit is uniform fertility. But in RCBD, we use in mostly in field conditions where our fertility of soil differ from one place to other place. So we use a randomization in RCBD.
Okay. Okay, thank you very much.
Okay.
Arpit, sir, kindly proceed it.
Thank you so much, Prabhas sir. You have chaired this session of Track 4 in this international conference. We are glad to have you. And all the presentees have presented their work. Some of the students have also presented their work. And they are also on working day and night. They used to work on their particular research. And people like you, Prabhas sir, always motivate these students so that they can grow more with the queries you have asked from them. So thank you so much, sir, for being in the session.
Thank you very much, sir.
Thank you. Thanks a lot, sir. Now we will move towards the valedictory part, the last part of the session. And for that, I would like to welcome Professor Ashwini Wow, ma'am. Ma'am, please initiate this process. Thank you.
Thank you, Arpit sir, for greatly coordinating this session. Thank you, respected session chair, Dr. Deepak Mishra sir, for commanding the session. Thank you, all of you. We will start our valedictory from 4 p.m. Up till now, we conclude this session. Is 4 p.m. we will start with valedictory with the same link. But now we will terminate this session. We will having the break. Thank you. All of you may join in 4 p.m. with the same link. Participants may leave the meeting and again join from 4 p.m. for the valedictory session. Thank you. Hello, Bimlesh sir.